Okay, well, welcome again, everyone. This is Reclaim meeting number 1 of 2023. Today, we'll be hearing from Fran Quero. We'll have a few Q&As after his presentation, and then we'll open up the floor to everyone else in the community to, if they have any questions or want to give any updates on what they're doing. It would be lovely to hear from you guys. So I want to introduce you to Fran. Sorry, Fran. Fran has quite an interdisciplinary background, but broadly his interests lie in bioinformatics, synthetic biology, molecular diagnostics, and open source science and communities. He's been involved with the Madrid IGEM teams in 2018 and 2019, and is now working on the DNA detective project at the Learning Planet Institute in Paris. He co-leads a research group on the new generation of nucleic acid amplification, so Quasar and LAMP, and include designing affordable, easy to build open source hardware fluorescent detector that can be applied at anywhere at low cost. Fran's group has been developing this new biosensing technology in low resource settings, and they've been actively working with local partners in Cameroon and Ghana to explore the best ways to manufacture and deploy the technology through open science solutions. So I shall now pass you on to Fran so you can hear more about his work firsthand. Thanks very much for joining us, Fran.
Thank you, Jan. Should I share the screen somehow? Here. Wow, I was finding the button. So thank you very much. Thank you for the invite. I am happy to be here in Reclon. It's a community that I am kind of linked because of the nature of the DNA detective project. We used to produce a lot of proteins, so a lot of enzyme, well, not a lot, but a bunch of it, and it's always linked with the protocols in the Reclon community. So in the Learning Planet Institute, together with my colleagues, we work in the DNA detective project that is centered in developing low resource settings DNA amplification technologies, and we basically work with LAMP, which is like the most standard common one. So I think that everyone, or because I think that people here comes either from Reclon or from the global LAMP group, but just as a quick introduction of LAMP, is an isothermal amplification, which means like it don't requires thermal cycling. That is basically, I mean, the key factors of the technology are basically two. One is the use of a special polymerase called BST polymerase that have the ability that around when the DNA is around 63 degrees, which means it's not completely tight, the polymerase have the strand displacement ability. So it's able to displace the double chain of the DNA to amplify the DNA that normally is done with a thermal cycler. So in this case, it's done thanks to the BST polymerase. And the second key feature is like the inclusion of special primers that have the ability to loop with itself. And this, well, the use of 6 primers, and 2 of them have the ability to loop with itself. So that's first fasten the amplification. Second is supposed to add the specificity. Anyway, like LAMP still struggle from the problems of false positives, but still it's supposed these 6 primers, the use of 6 primers, to add some specificity. And lastly, like it adds a lot of like reaction speed to the amplification. It's a technology that is quite old. Actually, like it was developed around the 2000, or the 2000. And so recent, like nowadays, there are a lot of variation of this technology. And well, this is a kind of graph trying to separate a bit or categorize a bit the different adaptations of LAMP based in two axes. One is like if the adaptation of LAMP are endpoint adaptations of real time, which means if you read the result at the end of the amplification, for example, at the end of a 60-minute duration, or if you read the signal, the fluorescent signal during the amplification. So you have like points of amplification signal during the entire 60 minutes or whatever of the amplification. The other like axis, this main one, the real time and endpoint, is important for the presentation of today. The other one that is less important is, because I am not going to center a bit the hardware on this part, is if the signal generation is dependent or independent of the amplification that was carried out. So simplifying it, it means like if, for example, the signal, if it's a color change or its fluorescence was just generated by a pH change, for example, which actually, I mean, independently of what's the amplification going on inside of the tube, this pH will shift anyway. Or if the signal is specific to the DNA that got amplified. And this you can do it with different methods. One of them is, for example, using like a specifically designed molecular probes that just generate the fluorescence when they recognize a specific amplified DNA. So the signal will just be generated when a specific DNA from coronavirus is amplified. I am not going to enter a lot on this because, as I say, this is kind of not important for the presentation, but there is a lot of different methods for addressing this. So I'm going to start. I'm going to divide the presentation a bit in two parts. One for the endpoint hardware, which means like the hardware that just serves for incubating the reactions and measure the signal at the end of the reaction. And in that sense, the reaction can be just basically can give out two results, positive results or negative results. And then after I will enter in the real-time hardware, which allow to measure like the reaction in real time and study the kinetics, etc. So I have been trying to work in two different hardware around this. The first one is like a kind of a water bath that is completely digitally manufactured. And the idea was that it was inexpensive. So you can make one water bath by 5–10 euros. And also the idea was to create a piece of hardware that is replicable. Because there are a lot of people around that hacking different things like kitchen stuff or whatever, like just resistor or buying in AliExpress, like inexpensive incubators. The idea was to do a replicable piece of hardware that is inexpensive and people can just reproduce and use it. So it's basically have two different buttons, like you just fill it with water, put the reactions inside, close the water bath with this kind of cap that have like is kind of an orange acrylic that after will serve as a filter for the light. So you basically put the reactions inside, press one button, the water bath heats to 63. You can adjust it inside with a potentiometer. And after 1 hour, when the amplification is supposed to have happened, you can press the other button and have some LEDs in the side. The chamber have some LEDs in the side. It lights the inside chamber and you can read the fluorescence of the tubes from the top. So, well, basically it's like that. We have some lyophilized reactions. You just pipe it through your rotation buffer and some sample, put it inside of the water bath, fill the water bath, put it inside, incubate during 60 minutes and read the fluorescent results from the top. So I have been comparing the results of this with a normal thermal cycler. I mean, without thermal cycling, just the thermal cycle is 63 degrees. And the results are quite the same after 1 hour. These are reactions for coronavirus. A lot of, so the limit of detection of the reactions at that moment, it was like 20 copies of coronavirus per reaction. Now we are trying to narrow a bit down, a bit lower, like to increase this limit of detection. But the comparison between the water bath and the thermal cycler seems to be quite replicable. So then the main problem with this device is like it was actually difficult to produce it. Mainly because making this wall where the LEDs are watertight, it was quite difficult. It was not quite difficult, but annoying. And it was not replicable. It was more an art. You need to put PDMS or silicon there. And I mean, people that are not like used to digital manufacturing or I don't know, silicon or whatever, it can become a bit tricky and a bit difficult and a bit messy. So I was thinking how to simplify this and make it like the simpler version of something that can incubate and read the reactions. And I came out with this design that now we are testing. I mean, it kind of works. I still have some problems of heat uniformity, but it will have in the next iteration, whenever I have time for working on this, it will have a simple solution. And basically it's a PCB with the holes for the tube strip and some resistor for heating. And these holes are kind of surrounded by the copper of the PCB. So it creates like a ring around the tubes that heats at 63 degrees. The circuit is the same than the water bath. The only thing that changes is not the circuit, it's more like it's just a PCB, not a water bath, don't involve water. The idea is that you put the tubes, you hit one button, incubate with at 63 during 1 hour, and then at the end you press the second button. There are LEDs, lateral LEDs there are lateral LEDs in the lateral of the tube, and then when you turn the PCB, there is one slit for a filter that surround the bottom of the tube, and you can measure, you can see the fluorescence. I don't have like real photos of this because as I say, I just received it like a couple of months ago, and I haven't had time for playing with real amplifications. I measured the temperature with water. There are 63 degrees replica, like there are stable 63 degrees here in these tubes. In the lateral of the entire PCB, it drops a bit. So it's something I need to solve. And also the other problem, I was talking yesterday with a colleague from the lab, with Kai, that I think is also here. It can be like also the problem of condensation, because you are just heating the reaction in this ring. So probably the entire like reaction will evaporate a bit and condensate on the top. For doing this, I think the simpler solution will be putting a bit of mineral oil on the top of the reaction to make it sealed and avoid the condensation problems. Mineral oil is cheap, is affordable, so shouldn't be a problem. So these are the two endpoint hardware I have been working with. And now I'm going to talk a bit about the real-time hardware, just for as a summary of why real-time can be interesting. There's a lot of different applications of real-time hardware. The most standard or common ones as an introduction are first for spotting like false positives in an amplification. And this can be done because if you just have the endpoint amplification in this course here, you just have a tube strip like this, 4 full positives, 3 complete negative, and 1 that is a bit like between positive or negative, as an example. And then if you just have this information at the end of the reaction, you don't know really what happened inside. But if you have been measuring the entire fluorescence through the amplification, you can actually see the amplification curve. One of them don't have a sigmoidal kinetic. And that normally means that something else than a DNA amplification happened. It can be a lot of things, and this is of course more complex than a simple like example, but having the kinetics of the reaction can help you spotting which of the signals were not generated by a DNA amplification. Amplification normally is exponential always and have a sigmoidal product. The second application of real-time hardware is for actually people trying to develop their own reactions or trying to produce their own protein. So having the kinetics allows you to, for example, put a threshold where when the reaction came positive, and then you can measure the time where each reaction, at the time of each of these conditions came positive. For example, if you are comparing different enzyme concentrations, you can do some kinetic curves. This is basically from the PCR. We are still wondering how robust is LAMP to do this. But in theory, if you have like the threshold, the time to threshold of each one of these conditions, you can make some kind of correlation between the enzyme units. So you can do a calibration curve. I will show after some results with commercial enzyme, and then you put your sample enzyme that you want to know the activity, and you can kind of correlate or know more or less how many units you have in your homemade produced enzyme. Or for example, if you are trying to analyze different primer sets to see which of them are faster, so you can have the kinetic curves and try to see. So before entering directly in the hardware, I want to also spend some slides here in talking about the process of developing the hardware, because it has been through a community kind of related with Reclone, or at least with the same spirit, which is GOSH, the gathering for open science hardware, that a bunch of people there in the community, we started having recently in the last 2 years, I would say, interest in developing open source hardware for real-time amplification, for real-time nucleic acid amplification. And this came either from the hardware point of view, in examples like Urs Gaudens from Gaudi Labs, that he already developed like the pocket PCR, like a thermal cycler for amplifying DNA. And now he were interested in moving through to real-time hardware, like a real-time pocket PCR, real-time pocket PCR, sorry. Or either the other example is, for example, me, that we came from the biology part, like we were doing LAMP and we wanted to have a device that can measure this LAMP in real time that is affordable and is open source. So let's say that from this both point of view, from the biology or from the hardware, we mix a community, we gather a community around developing some open source hardware for nucleic acid amplification. And thanks to those, thanks to the collaborative developing funds, we have the opportunity to gather here in Paris and experiment together, and came out with 3 prototypes. These were the super early versions of the 3 prototypes that we were working with. The OpenQ lamp is the one I will explain today. The webcam Q lamp is now the pocket qPCR of URSS. It's not yet in the market, it's not yet out there, but will come soon in the following months. And the Chile lamp that was baptized by URSS and Boris from Chile, from collaborating with Fran Federici, the lab de Tecnología Libre. And well, it's similar a bit in the technology to the OpenQ lamp. So we both, the Chile lamp and the OpenQ lamp, we both use analogic sensors. Meanwhile, URSS based the pocket qPCR in just putting a camera on the top of the tubes. Now it's in the lateral. In this prototype was on the top. I'm measuring, like taking images, I'm measuring, analyzing these images through the time for measuring fluorescence. So with these 3 prototypes, the second residence of GORS was based in Mboalaf in Cameroon, and we were trying to work with the local community in Mboalaf in a real case scenario. They were protein producers and they were doing lamps, so we were trying to see the feedback from them on the real world application. So and use this feedback for creating a second iteration of the hardware. So we spent there 10 days. It was an amazing time. We came with a lot of new ideas that we are actually still implementing in the second and third iterations. So just now, for entering directly in the real-time hardware, this is the current version of the Q lamp. It works. It works for 8 tube strip. The price of production price is around 40 euros, less than 50 euros, depending on the scale that you produce. It can be even lower. And we'll have exchangeable optics. So here there is a little PCB with the LEDs, but the entire hardware is made in a way that if you want to put different wavelengths, or probably in the future I will start experimenting adding different wavelengths to the same PCB. So it allows you to just order this PCB and have different PCBs and exchanging them depending on your experiment. It's based in USB Type-C with an ESP32, which gives Wi-Fi and I think Bluetooth, the one that I have added, not. I didn't experiment with Bluetooth, just with Wi-Fi, and works through USB Type-C, which means that can be powerable through power banks. So it gives some kind of also power supply independence in case that there is not a stable power supply available. I say that it works through Wi-Fi, but don't require internet. So you just need to be connected to the same local network with a computer or a phone and the device, but the local network don't require internet. So if you just have a smartphone, you can put your local network, even if the smartphone don't have like a internet connection, you can still connect the machine and a computer to the smartphone, and they communicate without the need of internet. So, well, the sensing unit is like the entire device can be resumed in this part of the sensing unit. There is a metal 3D printed block in aluminum that costs around 5 euros. And then the PCB have like lateral LEDs that light the chamber where the reaction is getting incubated. And then if the reaction is positive, the fluorescence goes through the bottom through a Lee filter that are super inexpensive filters. I mean, the price is almost nothing. It can be even, one A2, I think, costs like 3 euros. So with 3 euros, you have for 1 year, I don't know, more than 100 devices. And then there is a photodiode in the bottom that measures, generate a current signal that is processed through an analogical stage and is measured with with an analog digital converter that goes into the microchip. So the electronics in total cost, well, and then there's the heater in the lateral of the metal part that have like 3 different segments. So it's 2 tubes. It's 2 tubes with 1 tube of spacing. You can put, in theory, 1 different temperature. So this is made for, in the future, when I have time for recalibrating this, for being able to do gradient lamp. I didn't try it yet, but let's say it's also helping for maintaining the entire thermal block uniform. The difference in temperature between tubes is less than 0.1 degree. These have been measured with an external thermometer, so it makes kind of robust temperature equation. So I am going to show some of the results. They are preliminary results, but still seems good, the amplification curves. This first one, because we are in Reclone, the first results that I am trying to show is like the enzyme quantification. We were producing BST polymerase. So what we have done in this experiment, that probably needs to be done with more duplicates, but is to prepare a standard calibration curve with different concentrations of NEB enzyme. Probably we used a lot of NEB enzyme. This experiment should be done with much lower amounts, but there is just the calibration in purple, the calibration with different concentration of enzyme. And there is just a threshold value here, and I take the threshold value where the amplification with different concentration cut, and I do a correlation, exponential correlation between the inverse of the threshold, time to threshold, and the amount of units I add in that tube. So then you have the calibration curve, and you can put like 2 different concentrations of enzyme, and then kind of get at least with enough variance, like more or less, how many units approximately of BST you have in your tube. This can be useful, I feel, for all the independent or distributed, like independent local protein producers that wants to have a cheap and affordable way and simple way of quantifying the protein. I think that can be interesting to have a device that can be used for that without the need of a qPCR. Then this is more in the diagnostics or detection part. This is an amplification curve of coronavirus, RNA copies of coronavirus, but we have also tried with inactivated virus. And this is like, I plot the result here in a semi-quantitative way, which means like instead of having a complete quantitative model, I just put 3 groups, 200 RNA copies per microliter, 20 RNA copies per microliter, and 2 RNA copies per microliter. And I get the threshold values of the different amplifications, the time where they cut the threshold value. And so with duplicates, and it still have enough variance for differentiating the different concentrations. Still, this needs to be repeated. We need to try to figure out how many replicates, if these results maintain, etc. Just for you to see a bit of the results. And this is a trial of doing a more quantitative result, like of just getting the threshold values, the time to threshold value, and doing a quantitative regression, an inverse regression. And the regression I like because, for example, it have here an asymptote that is exactly 32.26, which means that even if you put like infinite amount of DNA in your reaction, the reaction will never happen in less than 32 minutes. This is what this parameter here indicates you. And also the other important thing is like the points below 3.1 nanograms per microliter, the reactions that have had less than that amount of DNA were having too much error compared to the regression value, that this could indicate you that actually this is your detection, your limit of detection of your reactions, because below this amount of DNA, the threshold value is correlated with other things that are not the DNA. For example, the occurrence of false positives or the statistical variance in your reaction, because you think that you are putting 2 chains of cinnamomi. This is like a pathogen for trees, sorry, I forgot to mention. But you think that you are putting 2 DNA copies, but actually you are putting 5, and this is because statistically the variance where you are putting too low amounts of DNA happen to be too big compared to what you are thinking you're putting. So anyway, like these points here, that goes when the DNA is too low, goes out of the regression, can indicate actually the limit of detection. And this helps you to do some kind of system biology of LAMP, where you can also modelize the reaction, understand how it works, and understand the variables that influence the reaction, etc. So yes, as I mentioned, this is like not for coronavirus. This is a pathogen infecting chestnut trees all over the world. So we're trying also to use this for the LAMP reactions and the hardware for agricultural purposes. Now we are trying to detect the pathogen instead of in the trees in the soil, which can be at the end the more useful thing, just to understand the quality of a soil instead of just the reaction is the pathogen is kind of obvious once that is infested the trees, but it can be interesting to detect it in the soil. So still in the diagnostics field, I have been also collaborating with Jenny Molloy, with Ana Pascual Garrigos from the Open Bioeconomy Lab. We were trying to also use this device for detecting in real time other kinds of diagnostics methods. The Cas12a, I think nowadays is one of the most popular ones, because I mean all the Swiss Institute, all the Wyss Institute and Harvard are trying to kind of patent a version of this called Sherlock. So it was interesting for us also to know if the device can be used for this technology. This just required to incubate the reaction from 37 degrees to until 63, because the Cas12 that they have is thermostable. The device can anyway, the range of temperatures for incubation is broad. So yeah, I mean, we were trying to incubate the reaction, measure the reaction in real time, and compare the results between a commercial TIC and a spectrophotometer and the device, and the results are kind of comparable. As you can see, the results are not sigmoidal, are not exponential, because this is not a DNA amplification. This is just an enzyme cutting fluorescent probes, but it doesn't amplify the DNA, so it's not like it amplify from one chain of DNA, it creates another one, and then it uses this new one plus the old one to create new ones, and it's exponential. No, this is just cutting in a linear way. So you can actually see in the kinetics, like compare this with LAMP and see like, okay, this actually makes sense, it's linear, it's not exponential. And then there is like 3 results outside of the diagnostics that I want to share. I have limited results around this, but I strongly believe that it's possible. So it's just as an idea to use the machine. Well, the first one is not actually an application, it's just a mode that I have implemented in the device. So it's like the melting curve mode. This is actually the melting curve from the experiment I showed before with the protein quantification. And it's just for showing the ability of the device to increase 0.2 degrees from 50 degrees to 90 degrees and measure the fluorescence in real time. So you can actually see where the double strand of the DNA separates. No, this is interesting because if you have false, it could be a way of spotting false positives, because when there is a false positive caused, for example, by primer dimers, the melting point, because the DNA sequence is way shorter, the melting point decreases. The DNA separates at lower temperatures. So this is just the same DNA. So the melting point is around 80, from 85 to 90, more or less the same. But if it was a false positive, this peak here of the melting point, when the DNA melts, it will be displaced to the left. Sorry, this here, I don't know why it didn't add it by error, but this is the y-axis is the differential, is the inverse of the differential of fluorescence divided by the differential of time. Is the standard melting curve metric. So this is just for showing that with this device it's possible to do melting curve, and now we are trying to use it for spotting false positives, or I mean, there are other applications. Probably there are people here in the call that this generates them some ideas on how to use this. But just in theory and in practically, it's possible to do melting curves with the device. And then there is the 2 last applications before ending the presentation. The first one is to do antimicrobial sensitivity test. And this, what we are doing, what I am trying to do in the lab, is to include like non-cell permeable intercallant dyes that when they enter in contact with DNA, they generate the signal. So I include this in the same solution than bacteria that are, for example, resistant to ampicillin or not resistant to ampicillin. And okay, if you include ampicillin, the bacteria that are not resistant, they will break and expose the DNA, and then because there is intercalum dye in the solution, this will generate a signal. If the bacteria is resistant to ampicillin, they won't break and the signal will not be generated. And you can measure this in time and see how the bacteria are dying or breaking because of the antibiotic or not, depending on if they are resistant or not, and then take conclusions about which resistance like the different bacteria have. I haven't tried this in the lab with antibiotics. What I did as a preliminary, I did just before the holidays, I just incubate the bacteria with heat for just breaking them and see if like this was working and the machine was sensitive enough for the fluorescence, and it worked. Like with temperature, I was increasing the temperature gradually, and at some point the bacteria were exploding because of the temperature, and they were generating the signal because of the DNA getting into the solution. This can be useful for antimicrobial resistance screening in low resource settings, for example. And the last one that hopefully we are going to try this soon, the next month, is use the machine for cell-free kinetics, cell-free protein production kinetics. So you can imagine to have different like synthetic genes constructions with different, for example, you have 8 different promoters, and you want to try which of the promoters are faster than the others. So you can include these 8, or I don't know, whatever construct in one of them in each tube in a cell-free extract, and you can bind in at the end of the promoter. You can produce GFP that generates a green fluorescence. So if you measure this production overnight, you can actually get a plot of how fast GFP is getting produced. So you can actually compare different constructs with this device. I mean, it's similar to what you will do in a Tican spectrophotometer, but in a machine that costs to produce 50 euros. So this, hopefully we got some lysate in the lab, and we have some DNA for GFP. So hopefully in 1 month, I will have some results. Also, we are going to collaborate with Fran, and we have a trip for working around this together. So hopefully we can do some trials in a couple of days, actually. So this is all from my side. I wanted to thank a bit all the team from my both PIs. Ariel is the director of the institute that I am there, as well as Jenny, that have been helping me since I was in my master in China during all the confinement and all the crazy pandemic. Both of them have been a huge support for all this work. And also to my both mentors, either in hardware, Urs Gaudens from Gaudi Labs, and from the biology from LAMP, a guy that is working with me now in the Learning Planet. And also to the rest of the team, the rest of the global LAMP community, the Mboa Lab team, and also the rest. I mean, Anna from Open Bioeconomy Lab that helped me with the CRISPR, and Hortense and Jan from the Learning Planet Institute that are my lab colleagues. This is all from my side. Here are the QR code for the Telegram group that we are kind of talking, me, Urs, a huge community around global LAMP, we call it. And this is the GitLab where there are the designs of the water bath and the QLAMP machine. This is still just in my local files. I need to upload it. And here is my email. Thank you very much. Sorry if it was a bit long.
No, that was perfect. Thank you very much, Fran. That was really interesting to hear from you. And I believe there's a few questions and comments in the chat.
Sorry, maybe someone wanted to take a photo. It's okay, it's recorded.
Yes, I can go over. So yes, we had a comment from Fernand saying that mineral oil is perfect. That they use it with guys GMO detective reactions. And then we have a comment and a question, some questions from Harris. I don't know, Harris, if you want to ask them directly or you prefer if I read them.
I can ask them directly if that's okay, because they might have changed a bit, but I'll try to not take too much time. Should I do it now or? Yeah, so to be honest, I'm a bit of a newbie in the open, like in Gauche, and I was in the Internet of IOPA meeting now. So I'm just discovering the open world. But I have a background in, well, in philosophy. So I was studying science. But when I was studying science and I was in physics, I was always thinking of high-level science being siloed away in universities. And what I saw today it was basically science of the last 20 years or less being produced, or being like somebody giving the blueprint and say, you can do this to the lay citizen. And I think, but the thing is, I don't think that many lay citizens, like many people who are not top scientists, know about this. So I'm wondering whether we should make an effort in, together with the hardware, in order to truly make science open, whether we should make a bit more of an effort in educational material. That's what's missing, because I guess the end point, or the goal, is to enable citizens to have more access to science. That's one. And a briefer one, which is a warrior of another hat that I wear with effective altruism, like a bunch of people, mostly in top universities, thinking about the future, which is what giving this to the people, isn't there a danger of baddies creating proteins, and what happens is, like, for very cheaply, like out of control, essentially. Like, again, it's the same idea of bringing the technology to the people. It's the same worry, basically, this other question. It's just the flip side of it. And yeah, I'll stop. I hope that was a bit longer, but
It's okay. Yeah, so I think that probably in this call, I am seeing people that are probably much more able to answer your question properly. My personal view is, like, when I started, this was actually, I started learning from hardware and DIY bio, I think with 17 years old, it was 10 years ago. From URSS, from, I don't know, also Guy, at some point, I also, like, the DNA detective project, I got involved, and at that moment it was even more difficult. I think now more and more there is, like, bigger and bigger documentation, people working from the academia, even in understanding this movement, mixing with people outside of the academia, and it's getting more and more diverse. And it's true that a lot of them are facing some of the problems that you say. For example, the biosafety, which is one of the main issues, right? Like, what happens when people can grow whatever, or can produce, can transform bacteria, and what happened with all these GMOs outside of the academia that before was controlled. I mean, it's a huge topic, have been addressed by people that have much more experience than me, and it's about, I feel it's about finding a compromise between being able to do outside of the academia, but still with biosafety and still control. And there's all this effort in making all the regulation that before was not existing. Places like IGEM, like the IGEM competition, I feel is doing a huge work around that. Like, there are teams, before the teams were just belonging to universities. Now there are community labs and biohacking space that are doing synthetic biology and presenting to IGEM. And I think inside of the IGEM community were one of the first times that these issues were faced. So there are these huge communities are trying to do it. To be honest, I think that I think there were, it's been a lot of progress in the recent years in regulate all of this and allow people decentralize a bit the biology and the research, but still in a safe way. Sorry if I did like. I am reading something from the Open Bioeconomy Lab, like, following on Harvey's question, are the open QLAM hardware specs available somewhere for people to make it themselves? Let me reshare 1 second the screen, because I think I closed it too fast. In this slide here, you can see the GitLab. With this QR code, if you scan it, you go to the GitLab of the Open Bioeconomy Lab, where the documentation of the Open QLAM hardware for making it and how to use it and everything, like, everything is documented here in the GitLab. So feel free to go there. Contact me if you have any question.
Oh, thank you. Yes, I just missed that, I think. And then we have another comment question from Aaron. Would you like to read that out, or do you want me to?
Yeah. So, hi, Fran. So, in our projects, we are actually trying to build some open source hardware too. So, we're actually curious if how much, if you have done the maths on how much we can save this, because it will kind of help us maybe in some kind of trying to apply for grants to like show them how much we can save on things like that. And do you think the component parts of this are like really available on the third world countries so much that we can build it on our own?
Right. So the first question, I understood it barely because I didn't understand part of it. But I mean, yeah, feel free to contact me after. I understood that you want to leverage in this hardware and to use this also for getting, I mean, applying to grants and showing that this open hardware have potential. I mean, feel free to contact me after this meeting. We can talk about it. I will be happy to help in any way. The second part, like how available are these components? So I put a lot of effort in trying to make the entire process easily reproducible for people in the way, for example, and also it was also useful for me for scaling the production of these devices. So at some point we want to start selling or whatever. It needs to take me, this I got a lot of inspiration from others, from Gaudi Labs. It needs to take me as less effort as possible to produce these devices. It cannot take me one device to produce one day, no? So, for example, the electronics, I already documented everything and put it in a platform, the open source hardware lab, in a platform that you can really, in 2 steps, order the device, the electronics already assembled. Because another thing that I did with electronics is trying to design them for industrial production. So the components are pretty small, which means that it's difficult to solder by hand. But for producing it in, for example, Shenzhen, is super cheap because it's exactly what the industries are used for. Like to solder, these robots are always used to solder these small surface components. So it have the problem that it's difficult to produce by hand, but it's super easy to order. And one unit, like if you buy just one PCB, it should cost you all the electronics around 40 euros, maybe a bit more expensive than if you buy 10 or 50, but still 40 euros for the electronics and still you 5 euros or 10 euros for the metal piece, plus the 3D printed case, I think is still possible. And yes, it's easy. Like I put a lot of focus in making it easy for buying, replicating. Sorry, I don't know if I answered the question. You can re-ask me.
Yes, thank you for answering my question. So, and we just to clarify, do you think it would be better to just buy it from you guys or so, or do you think
I mean, for sure it
to
will help us. So as I spent like 1 year full focus on the project, so I think it for sure will help us to continue. We are not selling it by the production price, so if we are producing it by 50 euros, probably we will sell it around 200 and 300, counting also. So we need to rent the space, the working hours, blah, 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 the shipping, all these things. If it's possible for you to buy it, you will also get from our side the help on. I mean, it will be the machine entirely assembly, working, functional, and we can do a follow-up in how the device works. Right now, it will take a few months for us to start selling it, I feel. So also depending, you can wait like a few months. Otherwise, it's all documentation. And I will also be happy if you have any doubt or whatever, like we can also see, because I am interested in other people replicating it. So let's talk about it. Just contact me after if you want. Here is my email. And yeah, let's talk.
Yeah, perfect. Thank you very much.
So we have another question in the chat from Leonard. Do you want to read it or shall I?
Of course. Can you hear me?
Yeah.
So these are now all for low throughput 8 reactions. Do you have any plans for high throughput, so 96 reactions, or would that in general be too inaccessible in your opinion?
Sorry, it cuts a bit. I think it's my internet because sometimes it gets a bit unstable. I understood if we have any plans for making it more high throughput, right? Like instead of 8 tubes 3, for example, making 96 well plate or something like that.
Yes, or would that in general be too inaccessible?
Right. So in terms of how many reactions you can use this device, we think it's affordable enough for if you need to do 16 reactions or maybe, I don't know, 20, 32, whatever. You maybe can use side by side 2 devices, 3 devices. Still, you can control each device independently. So it's also have the advantage of having different temperatures and so freedom. For bigger volumes, it was since the first moment in the table to try to do something like 96 well plates. So the problem of that is like it's much more difficult to ensure that the temperature, the fluorescence, the replicability across reaction it's in the boundaries of, for example, this device, like for example, in the temperature 0.1 degrees of difference between tubes. It's much more difficult when you have a 96 well plate device. So we wanted to do something simple from now, affordable. Meanwhile, we are still thinking about how to move this forward. Now that Urs, for example, were playing around with the cameras, maybe it's more interesting to, in a 96 well plate device, to have just a camera instead of 96 sensors. So I will say that yes, for increasing the volume, we have some ideas and some plans in mind. We use this as a pilot to learning about the technology before scaling to bigger volumes. In terms of producing it, of being able to the high throughput, like I mean, being able to produce a relevant volume of these devices for distribute them. So yes, like since the beginning, it was a bit of our focus, like to try to not make it as less handmade as possible. Like the entire thing have been kind of adapted for production in a way that for us, we just need to order the components, assembly them, and doing some quality control. But producing one of the devices and the components don't involve a lot of time on the person. So this really radically helps in the amount of volume that we can potentially handle in the future.
Oh, I think that's all the questions and comments that are in the chat. Thank you, Brian, also for sharing the URL. Does anyone else have any questions that they want to just voice out? Or if not, we can open the floor to some open floor discussion, see if there's anyone who wants to update anything.
Fran, do you think you can show like the GUI, like the website, like how it looks?
This
Or
was
it's hard?
completely, I was expecting to be in the CRI today. I am at my place. So everything was prepared to do to show it the key. I can.
Do you have any pictures, just how it looks? Maybe it's worth it to show people.
I did a screenshot yesterday with the computer at work, so I don't know if I have maybe in the GitLab. It's quite simple and it's a standalone, so it works in Chrome in Explorer. So you just connect to the machine, enter in a web page, and then it shows it's like a web page inside a store inside of the device. So you can connect with your phone, with Linux, with Windows, with whatever. It's independently. You don't need to install nothing. So I think I don't have that was a fail. Yeah, sorry. Well, I will probably put it like this afternoon here in the GitLab for you to see it because it was an error. Yeah, take a screenshot and show it here. Now it will be too complicated because I was thinking to download the files, enter. It's going to be a mess.
No worries. I mean, feel free to share on the forum as well, so we can all have a look later, or just email it to me and I will send it out to the rest of the group later. But it's been a lovely insight into kind of what we can do with open source hardware, and very much interested in looking at your GitLab webpage about all this, see if we can implement it at the OBL maybe. Would anyone else like to chime in with any updates from their side? If not, we may just wrap it up for the day. Jeremy's just asked questions. I'm just going to read out quickly. What skills would be needed to try building some of these?
Sorry, I was writing the answer. I think that mainly time. I mean, it's not for designing this or for understanding the electronics. Mainly, I think that this analytical electronics can be a bit tricky. As far as we have been, the global LAMP team, we have been playing around with this for you. If you don't want to enter in the details of the electronics and just want to produce it, it's relatively simple. I have here in the GitLab, just for showing a bit of the screenshot of the GitLab. This is like the flow chart for producing one device. So you can just order the 3D printed parts, or you can just order 3D printed filament, go into a maker lab, print the case. You can order, probably you will want to order a 3D metal holder. All the files of the production files are in the GitLab. If you have any doubt, I feel like you can ask me, probably, like I can guide you a bit, but it's just sending the files to be produced, buying some leaf filters, buying the PCBs. Still, the PCBs, you just need to upload the files to a web page like PCBWay or whatever, and then just receive the things and assembly. And for assembly, it's quite straightforward. Connect it to the computer, upload the code through the Arduino IDE, and then I think that the rest is calibrating it for your research. So it's not like it's not super difficult, I feel. So I don't think that you need to know about electronics. Maybe a bit of digital manufacturing, a bit of 3D printing, probably, but it's quite straightforward, I feel.
Jeremy, you also had a follow-up question. Would you like to say it yourself, or?
Yeah, so thanks. So yeah, I do have a bit of knowledge on digital fabrication, so that'd be pretty cool. Is there some sort of checklist as a follow-on of or a curated list of projects that presumably you could build yourself and build a lab from the ground up, or fully equip a lab?
I feel still, I think that here in the big guys, I feel the big people in this topic are actually in the call. I think that I saw some of them that knows much more. I am actually learning from them. So, but for me, I think it's a bit of, I learn a bit from Hacktivia. Hacktivia have a lot of like guides on how to build DIY lab equipment. Now there is a lot of YouTube channels. Like there is actually the Thought Emporium, I think, that is doing a kind of videos about this recently. Sebastian Kosiova, there is a bunch of places. I think that you can know more about it through the Reclone and the gathering of open science hardware. Specifically about lab equipment, I think the Gauche forum can serve you as a starting point for asking in the forum and get help. Yeah, Hacktivia, Brian is showing sharing the Hacktivia link. This and Gaudi Labs is where I learned. Like when I was a teenager, Urs was one of the first person doing this, and I was just learning from him. Like he was uploading all these blueprints and the things. Probably nowadays it may be easier, the gathering of Ghost, gathering of open source hardware. I am writing it in the group. Yeah, there is an entire forum where all the people more or less doing scientific hardware is around there, like Hacktivia people, Gaudi Labs.
Okay, it looks like there's a fair few communities out there that we can learn from as well. It's still relatively new to me as well, so good to see that there are these many groups out there. But yeah, having a checklist, like one checklist on Reclone, could be also useful for people to just link through as well. So we should definitely try and implement that. Any more final thoughts before we close up for the day then? I would just like to point you to the 2 links that Sibeli posted. So as you know, we're rebooting Reclone, so we want to know your thoughts about how you want these community meetings to run, what you want to see. So feel free to, please do fill in our survey on that. And if you haven't already joined, do join the Reclin forum as well. Thank you very much. Saveli is asking me also, it would be lovely to see everyone's faces and just take a photo and share it around to the group. So if you all don't mind, please do open up your cameras. I think you're still sharing screen, Fran. If we can, yeah, perfect. Okay. Thank you for indulging us. It's always nice to see friendly faces and when we're having these meetings. And I know it's quite late for some of you guys. We'll wait a couple more seconds, see if anyone else wants to open their cameras. If not, then smile. Okay, great. I've taken a couple of screenshots and I'll share it around in the next email. I just want to thank Fran once again for sharing about all the open QLAMP, and everyone for joining us. See you guys next time. Bye now.